Good evening, everyone. Welcome you all to today's Week 10 NPTEL live session, which is on RNA biology. Okay, so today also we are going to discuss few questions which will help you to do the assignments, and I hope that my screen is visible to everyone and my voice is audible. If you have any issue with this screen and voice, then you can just tell me, okay? Write the thing in the chat box. Also, we are going to discuss a few questions based on non-coding RNAs. So, if you have any doubt regarding that time also, you can ask me, okay? So people are joining, so let's wait for 3 minutes. We are going to start the session at 6:10. Okay. Okay, let's start then. So first of all, let's discuss about the things we have discussed in last session. So it will help us to understand the NC non-coding RNA related modifications and issues will and we'll understand it better. So last class we discussed about genetic imprinting. So what is imprinting? It is basically we can call it as a gene silencing process. So in this gene silencing process, we are getting two alleles of every gene. Okay, every human being and every mammal has two copies of a single gene. One copy is from inherited from mother and another copy inherited from father. Okay, and these two copies are called allele. One gene has two allele. Sometimes in most of the cases what happens, only a single allele of a gene gets expressed while other allele of the gene get repressed or get silenced. So that in that case, in that particular organism, only a single copy of gene, that is, it may be maternal or it may be paternal, it will show its effect. If, for example, if it is maternal gene, then it will be showing most inherited characteristics from the maternal side, not the paternal side. Okay, so this is called generally genetic imprinting, and it has important role in many of the fetus development and size of where we understand that different size of animals are there. So size of animals also get modified because of this genetic imprinting, and also we are going to discuss about few more examples. So this is just a basic background about genetic imprinting. Okay, so, and next is this process is somewhat similar to doses compensation. So next thing we discussed in last class was doses compensation. So what is doses compensation? As the name suggests, when there are, it happens to the sex chromosomes. Okay, in case of humans, what happen, females have two X chromosomes, but male has one X chromosome and another one is Y chromosome. So in that case, if the female produce products from two X chromosomes, then it will be highly expressed in case of females as comparison to male, because it has one X chromosome. To balance, to these effects or balance the X chromosome products in both male and female, so here comes your doses compensation, where one X chromosome of the female body gets silenced or repressed. Okay, using which RNA helps in this X chromosome or doses compensation or X chromosome silencing. Basically we talked about XIST, right? XIST and T6, these are two antagonist RNAs. They regulate this X chromosome compensation or doses compensation in case of mammals, also in different organisms. For example, in C. elegans, Drosophila, we talked about this also. Okay, so now you understand what is doses compensation. It basically happens to the X chromosome when one X chromosome is more in case of male or female, then that needs to be repressed to balance out the products between two individuals, that means uh, between two different sex like male and female. Understood? Next is the difference of expression from both alleles are due to different states of chromatin, that is euchromatin and heterochromatin. Everyone understands the difference between euchromatin and heterochromatin. Okay, euchromatin is highly expressed while heterochromatin is, it is just like condensed chromatin. Okay, and also differential methylation of DNA. So what is methylation, acetylation? These are all epigenetic modifications, and everyone knows, and we also discussed in case of we were discussing about doses compensation, there are different types of epigenetic modification, including your methylation, acetylation, which regulate the uh, X chromosome uh, condensation. Okay, so in case of X chromosome condensation, we talked about that H3K9, that is your histone modification is highly essential. Understood? Histone modification, also there is one particular type of histone, a particular isoform of histone which is highly responsible for this. Okay, so this methylation also plays crucial role in X chromosome condensation. Okay, these are all about doses compensation and genetic imprinting. Next is your activity of small heterochromatic RNA, that is shRNA, appear to be essential for establishing and maintaining the imprinted status of genes. So what is this imprinted status? That means, for example, in your embryo state, one paternal chromosome get repressed. Okay, that needs to be maintained throughout the whole life process of that organism. It's not that it will get repressed up to the age of 10 or 12. After that the paternal chromosome will get activated and maternal will get repressed. This doesn't happen. Okay, everything just needs to be constant for the organism for the whole life. So for that case, these small heterochromatic RNAs, they maintain this repression, they maintain the imprinting, that if there is one chromosome which get imprinted, that need to be maintained for the whole life process. That is called the imprinted status of gene, that need to be maintained, and these non-coding RNAs are essential for that process. Understood? Next is the activity of various small heterochromatic RNAs It is not limited only to genetic imprinting, they have many roles. For example, translation, transcription, also protein modification. Okay, so this is a brief about the function of small heterochromatic RNA, and we are going to do questions on the transcription modification, translational modification, and protein modulation by this shRNAs. So let's start the question. You can write your answer in the chat box, then we'll discuss each and every question. Okay. So here is your first question. shRNA, that is small heterochromatic RNA, helps in chromatin condensation in RNA recognition model by interfering with replication, interacting with the expressed gene, binding with the nascent transcript, none. Which one is it? Please try to answer this question. Write the answer in the chat box, then we'll discuss it. Okay, let's discuss. It's okay if you are not sure. We are going to discuss it. So no problem. You can guess it anytime. Okay. So let's see. So your question is shRNA helps in chromatin condensation in RNA recognition model. So when this chromatin condensation happens, so here we just talked a few minutes before that genetic imprinting. Here one chromosome gets repressed. Also, while doses compensation, one sex chromosome, that is your X chromosome, gets repressed in case of mammals. So what is this repression? Generally chromosome condensation happens. Condensation means, if you remember the last class, we talked about it, that the length of chromosome gets decreased and decreased, and this X chromosome become act as a bar body. Okay, this bar body means it is highly condensed chromosome. It cannot be it cannot be expressed further in the whole life process of that organism. That acts as a bar body. Okay, so in that for this long chromosome to move it to transform into a bar body, there are multiple processes been happening at that time. And here this shRNA mediated chromosome condensation has been shown. There are two models are here. One is your DNA recognition model. That means when shRNA interacts with DNA to silence that particular gene. Okay, another one is your RNA recognition model. That means when your small heterochromatic RNA gets interaction with your RNA molecule to silence the particular gene at a RNA level. Here we are talking about at DNA level, and here it is RNA level. Okay, so here this shRNA, when it is we are talking about a DNA recognition model, that means it is recognizing a DNA, it must be complementary to the target complex. Okay, as you can see here, this is your target complex, that is yellow in color. This is your small heterochromatic RNA, and if you see here, it is complementary to certain base pairs, that is 20 to 22 nucleotides of this target complex. So in that case, this shRNA will come here and form base pairing, and after that base pairing, this is a double stranded DNA right now, and this unwanted double stranded DNA interact with this silencing factor, histone modifying enzyme, followed by silencing of that particular gene. Okay, this is not a single process, there are multiple processes involved, but how shRNA interact, that is the question here. So in case of DNA recognition model, it pairs with the template strand of the target complex. Here we want to repress this particular gene, then it will form double stranded base pairing, and that will lead to silencing of that particular gene. Okay, but this happens during replication, but here we are talking about RNA recognition. So what is RNA recognition? In case of RNA recognition model, the shRNA cannot interfere with the RNA while transcription process, and to suppress the gene of interest in case of RNA recognition model, it needs to be expressed fast. Okay, what is it? For example, this is your double stranded DNA, one is template strand, and from the template strand you are getting nascent transcript. I think everyone understand nascent transcript. Nascent transcript means when transcription happens and there will be one RNA molecule. Okay, there will be one mRNA molecule, but it has not gone under any type of post transcriptional modification. That is called nascent transcript. That means it has only 5 days to 3 days strand, or 3 days to 5 days strand, whatever it is. You can call it from left to right or right to left, but this is called nascent transcript. That means there is no cap or tail, that is nascent. This nascent transcript then undergoes post transcriptional modification, that is your capping process at one end. This is your capping. Another one is tailing. What is tailing? Tailing means adding of adenosine molecules. Okay, adenosines, that is tailing process. So this capping and tailing happens, that will be, it will be called as mature mRNA. Okay, mature mRNA or poly A tail. This mature mRNA now gets expressed, that means the gene is going to express, moving towards translation process. Now this shRNA comes, it will form a double strand with this particular region where this particular gene is there, which we need to silence it. This is your target complex. Now this targeting complex, silencing factor, histone modifying enzyme, and your shRNA comes here. they form a double-stranded base pairing with your target gene, and that will lead to suppression of that particular gene, or we can call it as including chromatin condensation. Okay, so here you understand two types of chromatin condensation which is initiated by shRNA. One is DNA recognition model. Here it directly interferes with the replication, but here in RNA recognition model it binds to the mature mRNA. That means when the gene gets expressed, then only this shRNA can bind to the mature transcript and form a double stranded RNA, followed by the suppression and chromatin condensation. Understood? So here is your answer: interacting with expressed gene. In case of RNA recognition model, it need to be interacting with expressed gene, not replication, not nascent transcript. Okay, fine. Let's move to next question then. So this was, we are talking about chromatin condensation, how shRNA helps in chromatin condensation. Let's move to our translation process. So translational regulation by this non-coding RNAs is achieved by antisense mechanism. Is it true or false? Here is a statement about translational regulation by non-coding RNA. Is it true or false? Okay, let's see. So we are talking about transcriptional regulation. Now it is translation. Translation means when RNA, this mRNA, post after ongoing the post transcriptional modification, moves to cytoplasm where ribosomes help in formation of proteins. using tRNA and other protein molecules or other enzymes. Okay, so this translational regulation by non-coding RNAs is achieved by antisense mechanism. What is this antisense mechanism? When non-coding RNA binds to target mRNA. One example is RNA interference or RNAi. Okay, let's talk about it. So here you see here that this is one example of antisense RNA downregulation. Okay, this is the translation of human HFE gene. Fe stands for iron. So this gene specifically regulates the iron balance inside the body. That HFE gene is downregulated by antisense RNA. Okay, so sometimes we need to upregulate a particular gene and sometimes we need to downregulate a particular gene. So this non-coding RNA helps in downregulate of this HFE gene using a antisense mechanism. So what is it? If you see here, this is the gene, okay, and it has this yellow color ones are these exons are, and these are antisense exons. Okay, so antisense means there are two strands. One while this yellow part is showing as the exons, that is sense exons. So as the name suggests, this is 5' to 3' region. Okay, 5' to 3' region, that is we can call it as a sense strand, while the opposite is the antisense strand. That's why the promoter stands from the 5' region. And if you see, this is your particular gene. Got it? Now let's move to transcription process. So in this transcription process, this HFE mRNA, when this promoter stands from your sense strand, okay, 5' to 3', it will produce the HFE mRNA, which will produce the protein using ribosomes. Done. This is a normal mechanism. But when after this when this antisense RNA gets activated, that means it will produce a particular RNA where this antisense exons, if you see here, if I draw here, this particular part of this antisense exon is complementary to the sense exon. Also this particular part is complementary to the sense exon. That means they are complementary, means they can form base pairing. Got it? So when this antisense promoter starts here for the transcription process, this is the antisense RNA where we are observing that this particular region is complementary to the sense exon. That's why they are forming a double-stranded RNA structure. Also this is a loop because this is not base pairing, this is not complementary, that's why it is forming a hairpin loop, but this is complementary. Okay, if see here, the opposite ones are, I highlighted those parts. So that in that case, this double-stranded RNA will not undergo translation process by ribosome. Only the single-stranded RNA, HFE mRNA, will undergo translation process by the ribosome to produce the protein. But if this is a double-stranded RNA, that will hamper the translation process, that will stop the translation process. Okay, understood? That's why the ribosome translation process got hampered and it will not produce any protein. Got it? So if you remember, this is one type of mechanism to stop the transcription process also. That one is row independent. Okay, in case of row independent, I am just trying to recall you some parts of molecular biology where which talked about in the mRNA, the RNA transcription process. Okay, during this RNA transcription process, there is a particular time where you need to terminate this process. So in case of this termination process, there are two types of things, like is row independent and row dependent. I am talking about row independent. In that case, while there is GC rich regions, that is guanine and cytosine, that form this hairpin like structure. Okay, now these are GC rich region which form the double bond structure. Now this protein machinery or enzyme machinery, which is just which is helping the in the transcription process, cannot move or cannot pass across this double stranded structure of RNA. That's why it will lead to termination of that particular transcription process. Here also we are observing the same thing. Using this antisense RNA, double stranded RNA has been formed so that it will just restrict the translation and restrict the protein production. Okay, I think everyone got it, that how this antisense RNA helping in gene down regulation, and this is also by non-coding RNAs. Okay, this is true. Understood? Okay, let's move to next question. Generally, non-coding RNAs target dash proteins to modulate protein function. Earlier we have been talking about protein production. Now I am talking about protein modulation of function. Do they target the nuclear proteins or cytoplasmic or both type of proteins or none? Which one is it? Try to answer this. Okay, so let's see what is it. So this non-coding RNA, what they target, which type of proteins they target to modulate a protein function. So if you want to modulate a protein function, just think it this way. Okay, if you want to modulate a particular protein function, so what will you do? I want to modulate, for example, X protein. Okay, so which type of protein I will target? Nuclear proteins or cytoplasmic? If you want to just change the function of this particular protein, then so how come this protein function gets modulated? See, the process of central dogma is replication, that is from DNA to DNA, then is transcription, that is DNA to RNA, and then RNA to protein, that is your translation process. Okay, and here we are talking about protein function modulation. That means we can easily do it when we just modulate the gene expression. Okay, for example, if I am having this X protein and I want to modulate its function, then if I just change the gene expression, okay, change the code of mRNA, then obviously the function will get changed. We don't need to interfere with any type of cytoplasmic and other kind of things. Don't need to do. We want to change the function. We don't want to change any type of location or other things. Okay, so here some non-coding RNAs can bind directly to proteins. Okay, we have also discussed about non-coding RNAs being bound to proteins. Okay, altering their structure, enzymatic activities, or ligand binding. Okay, so sometimes what happens, non-coding RNA just act as a string, and here different protein segments just bind like this. Okay, like beads on a string. And in this way, this particular non-coding RNA helps in modulation of this particular proteins. Okay, and this is specific to specific organisms and different cells. Got it? It is not ubiquitous for every cell. It is specific towards different cell. Next, targets of such non-coding RNAs often are proteins involved in transcription. Why they target the proteins involved in transcription? For example, nuclear receptors or general transcription factors. Everyone knows there are these transcription factors, replication factors. These all are proteins. For example, I want to just stop this function of X protein. Then I'll just try to modulate a particular protein or transcription factor so that the protein production will get suppressed, or the gene expression will get suppressed. Then there will be no protein. Okay, and if somehow you want to highly upregulate the function of a protein, then just highly express the gene, upregulate the expression of the gene, then the protein production will get upregulated. In that way you can manage the protein function modulation using non-coding RNAs. Okay, every time remember, when we are talking about some core changes like function or like some core changes, then we need to go for genetic level, not a protein level. So you just go for the genetic expression change, then everything will be changed. So here is your answer, that is nuclear. Okay, generally they target the nuclear proteins to modulate protein function. And this is one good example of protein function modulation, how it happens using 6S RNA. This is a non-coding RNA, and S70 function in E. coli. Okay, so this E. coli is a bacteria. So everyone knows about E. coli, I understand. So when there is a bacteria growth, there are three phases. Three growth phases of bacteria. One is your inoculation phase. Okay, when you inoculate a particular bacteria to the broth or medium. Okay, after that it will move to lag. First one is lag phase, log phase, then stationary phase. Okay, so lag, log and stationary phase. During the log phase, it is the exponential phase. That means if you get a curve like this, so this will be your lag phase. Okay, so this exponential growth you are seeing, this will be your log phase. Okay, so in this log phase, as you see, the growth of bacteria increasing, the number of bacteria are increasing, growth happening. So in that case, for the growth of bacteria, there need to be a number of proteins should be there, number of genes should be expressed, and for that different enzymes are responsible. So everything is required. Okay, that will lead to production of a lot of extra products. So this S70 function is there. We want to talk about only one function, that is S70, and this is RNA polymerase. When they bind with each other, they will lead to this is a kind of promoter. Okay, this promoter region, it is promoter, and it will just transcription will happen, and there will be a number of gene products will be there. Understood? This is about log phase, that is normal. But when they achieve the stationary phase is this. Okay, this is stationary phase. So in this stationary phase, the bacteria number get startled. They just stop like increasing their number or growth. So in this stationary phase, they don't need any extra products. They don't need any extra enzyme or any type of proteins to increase their number. Okay, in that case we need to suppress this particular action. How does this happen? Using non-coding RNA. This is the 6s RNA. Now it will bind to this particular interaction phase and will not so just suppress or just prevent the gene expression here. Got it? Now it will look like this. It will just prevent the gene expression where number of products get produced. Now this will look like this, and the gene protein production gets stopped because of this, or you can call it as S70 function. This S70 function gets stopped because of this 6s RNA, non-coding RNA. Okay, so this is one very good example of your protein function modulation using non-coding RNA. Okay, here also you are observing that they are interacting with your molecules like RNA polymerase is there. That means we are talking about transcription interaction. Okay, in case of transcription level, that is proteins in nucleus, not in cytoplasm. So generally they are interacting with the nuclear proteins, that is transcription factors, to stop this process in case of stationary phase, but allowed this process to happen in case of log phase. Understood? I hope everyone got this, how this protein function get modulated by non-coding RNAs. Okay, so here is the next question. RNAi is widespread among prokaryotes, eukaryotes, viruses, and all. Which one is it? RNAi, that is RNA interference. Okay, just a few minutes before we talked about RNAi, how this antisense mechanism regulate the expression of a particular gene. So this is widespread among what? Which organism? Yes, write your answer in the chat box. Then we'll discuss. Okay, let's see. So, this RNA interference, you must have heard about this term about when we call or when we remember about gene expression or gene cloning, genetic engineering. In everything genetic engineering or genome editing, this RNAi is quite common and widespread among eukaryotes. Okay, when we are talking about any plant genome modification or plant genome editing for drought resistance or for pesticide resistance or kind of or different types of defense mechanism. In that case we need to go for this type of RNAi or RNA interference. This specifically leads to a double stranded RNA formation which silence this particular target gene. Okay, let's see. RNAi is widespread among eukaryotes and highly evolutionarily conserved property and must have important functions. So why this it is related to evolution? Because it just um, relates its theory to first RNA world. Okay, in the starting of this particular class, that is RNA biology, in the session 1 and 2 we talked about RNA world, how this early world, like when there is the life been happening inside the ocean and there was nothing like any environment or nothing, how this small organic molecules get generated using this RNA molecule. We talked about ribozymes also, which has the highest catalytic activity during that time, and this double stranded RNA formation then lead to different types of nucleic acids like DNA and then proteins and then small structures like different living organisms that further evolution happen and lead to production of different higher organisms or complex structures or complex eukaryotic organisms. Okay, that's why it has evolutionary importance and it must be used. This important function can be defense mechanism against double stranded RNA containing viruses. So there are specific viruses which contain double stranded RNA as their genetic material. So it helps in this defense mechanism against this double stranded RNA containing virus. How they do this? By complementing, they just this RNA interference non-coding RNA, this will form a complementary strand to the double stranded RNA of this particular virus. Then the RNA enzyme will decode this, like yes, it is the site where we need to cut, then it will lead to cleavage of that particular RNA, that means destruction. Okay, we are going to talk about the function and what is the mechanism. Okay, no worry. It may stabilize the genome by sequestering repetitive sequences such as mobile genetic elements. Okay, so mobile genetic elements, or we can call it as transposons, that is lines and signs, long interspersed non-coding sequences like that. So they also help in stabilize the genome by sequestering the repetitive sequences. Okay, and control cellular development. Dicer knockout mice don't survive past gastrulation. Control cellular development. This is one important function of this RNAi, we can call it. That means we are talking about non-coding RNA. Okay, this non-coding RNA and how it functions inside a mammal cell or eukaryotic organism. So it helps in cellular development. This is one double RNase enzyme. Okay, dicer is one RNase enzyme. When it has been found that when there is a knockout of this dicer knockout mice, that means when there is a mice, we knock out this particular dicer enzyme, then the mice don't survive after the gastrulation period. So from this experiment it has been confirmed that it regulates the cellular development process. What is development process? That is from zygote to then moving towards embryo, then fetus, then a whole organism. Okay, this is the development process. It also helps in that case also. Let's see. Okay, now you understood that it has important function for eukaryotes. Next, this is another question about non-coding RNA. So, small interspersed interfering RNA, that is siRNA, replication happens in the nucleus of mammals. Is it true or false? Yes, answer this question. Okay, let's discuss it. Okay, RNAi is found in eukaryotes including fungi, plants and worms, or Drosophila and mammals. So this leads to this is the systemic nature of silencing in case of this fungi, plants and worms, and in case of Drosophila and mammals, that is higher eukaryotic organisms, it includes cell, that is autonomous silencing. Okay, next thing is it is heritable in case of fungi, plants and worms, but it is non-heritable from by Drosophila and mammals, that means in eukaryotes. And it can replicate siRNA with RNA dependent RNA polymerase, but no indication of siRNA replication in case of higher eukaryotic organisms. Why there is no replication of siRNA in mammals? Because of the absence of RNA dependent RNA polymerase, that is RDRP. Okay, we have DNA dependent RNA polymerase inside this Drosophila and mammals. We have DNA dependent polymerases, not RNA dependent polymerase. This is specific towards different viruses and other worms and uh, this worms and fungi. Okay, this is specific towards them. This is not found, this RDRP is not found in mammals, so there is no indication of siRNA replication. And what is the meaning of non-heritable? Non-heritable means each and every cell must produce their RNAi, that is RNA interfering non-coding RNA, for activation or for the functioning of the particular gene. Okay, it is non-heritable. That means if there is it is present inside the maternal copy or paternal copy, then it will not get heritable because there is no replication of this particular RNA. Okay, that's why. And for the activation or any functioning related to this siRNA, we need the cell needs to produce it. So siRNA replication happens in the nucleus of mammals. This is false, because mammals do not have RDRP, that is RNA dependent RNA polymerase. It is specific towards viruses and other organisms like fungi and worms. Next, which one is essential in miRNA mediated translational repression? Dicer, RISC, risk factor, double stranded RNA, or all? Okay, let's discuss. So, we are talking about translational repression by miRNA or siRNA, that is microRNA or small interfering RNA. See, here is the schematic representation where we can talk about both the function, that is RNAi and miRNA. So, for that case, this is one miRNA precursor and or double stranded RNA is required, and after that this will lead to uh, interaction with the dicer. Dicer is an endoribonuclease and it has evolutionary role. It is very important during your developmental processes and also helps in different small RNAs formation, okay, non-coding RNA formation. This is one endoribonuclease, this is one enzyme, okay. And this double stranded miRNA siRNA is here. You can talk, I am talking about this part or this part, whichever it is, no issue, okay. That will followed by the risk factor. So what is risk? RISC, it is also important for this small RNA production, and the full form of RISC risk is RNA induced silencing complex, okay, and it is a multi-protein complex which incorporates a one strand of small interfering RNA or microRNA. So okay, so this is one strand of, this may be, see, this is a double stranded miRNA and siRNA. It is using one strand. Here it is using this strand. So it is using this one strand of the double stranded RNA, which is miRNA or siRNA, as a template to recognize a complementary mRNA. Got it? Now it acts as a template. So here we are talking about, C. This is your mRNA, okay? This is your mRNA. and we are talking about translational repression. That means we want to silence a particular target target gene. In that case, this siRNA which we got here, that is act as a template, it will form its complementary base pairing in this particular mRNA. So if you see here, this siRNA needs perfect base pairing. That means these 20 nucleotides have been perfectly base paired with the mRNA or target gene. But in case of miRNA, you can see here, it doesn't require perfect base pairing. It just we need to find out the particular target, and some base pairing is enough. That is incomplete base pairing. Okay. In case of miRNA, incomplete base pairing is okay, but in case of siRNA, we need perfect base pairing for target cleavage. How this target cleavage happens? When there is a double-stranded RNA inside your cell, there must be this RNase enzyme. Okay. RNase enzyme. This RNase enzyme will cleave the double-stranded RNA. That means your translational repression will happen. In case of miRNA, same thing will happen, that is incomplete base pairing. As you can see here, this base pairing has been incomplete. That will lead to translation repression of a particular target gene which is on the mRNA but at a different site. So now you understand the difference between siRNA mediated translational repression and miRNA mediated translational repression. Here we are just cleaving a double stranded RNA in case of siRNA, but miRNA there is no cleavage or no destruction of the RNA. After binding or after forming the double stranded RNA, it will down regulate the gene which is present adjacent to it. Got it? And two important factors, that is dicer, one endoribonuclease, another one is risk. Why it is important? Because it will cut out this particular sequence. Okay. And another one is risk factor. This is the ribonucleoprotein complex, or we can call it multiprotein complex. That is RNA induced silencing complex. Understood? Next is, let's talk about this. MicroRNAs or miRNAs are not perfectly complementary to their targets. Okay, we saw that miRNAs do not induce target cleavage but block translation by binding to complementary mRNAs. Okay, so it just bind the complementary mRNA and block the translation process. It don't move towards the destruction or cleavage of the mRNA. But in case of siRNA, cleavage of mRNA happens. miRNAs are encoded by the host genome, whereas siRNAs in most cases originate from outer source. Understood? So miRNAs are encoded by the host genome. So microRNAs. So how this microRNAs get generated? Using this dicer. Okay, dicer has an important role in this microRNA generation. Whereas siRNA, most cases outer source means it may be from different organism also, which we use in genome editing or gene cloning or genetic engineering. We can include siRNA. Okay, these are generally 20 to 22 nucleotide, so it is easy to handle them, no issue. Got it? So which one is essential for miRNA mediated translational repression? So your answer is all. Everything is important. Dicer is a endoribonuclease, RISC is a RNA induced silencing complex, double stranded RNA which will lead to cleavage in case of siRNA, but in case of miRNA it will just lead to translational repression. I think it is clear to everyone. Is it clear? This is one important topic, okay, where you understand the mechanism of translational repression or the mechanism of RNAi, how it function. Is it clear to everyone? Okay. So is RNAi exclusively limited to cytoplasm or post-transcriptional control? Okay, so let's see. Although this is a very common view, it doesn't always have to be the case. Fine. And siRNA can be transported to nucleus and act as this small heterochromatic RNA to block the transcription. Now you got another point of siRNA. Okay. I think we talked about shRNA enough, where this shRNA helps in transcriptional regulation, translational regulation by blocking transcription and all from the beginning of this session. Now siRNA can be transported to the the nucleus to act as a shRNA. Okay, so issue is solved. Okay, so here is the next question. So long non-coding RNA, that is LNC RNA, helps in genomic imprinting. Is it true or false? You can just try to remember the previous session, so it will help you understand or answer this question. This LNC RNA helps in genomic imprinting. Okay, let's discuss about it. First thing, genomic imprinting, we have discussed it enough. That is the silencing of a paternal or maternal copy of the gene. That is, it may be a single allele from a particular gene. Okay, and two dominating mechanisms have been described for mediating imprinting in clusters. One is your insulator model. Another one is LNC RNA model. So this insulator model is evolutionary ancient but less utilized, and it is employed by H19 IZF2 imprinted locus, that is H19 LNC RNA in maternal expression. Okay, and this LNC RNA model is recently evolved and more commonly utilized mechanism. It is employed for non-coding RNA mediated imprinting at IZF2R locus, that is differentially methylated ICR. Okay, why this methylation is important? Because when we are talking about a chromatin condensation, okay, so everyone knows this histone is the important protein which helps in uh, just wrapping the long DNA structure. Okay, histone helps in form the chromosome, and when we are talking about histone methylation or acetylation, that leads to condensation or decondensation of your chromosome. Like for example, in your S phase or in your mitosis phase, when there is cell division, the chromosomes are highly condensed so that the cell metaphase stage can be properly happen. Okay, so in that case, this histone methylation and acetylation, that is epigenetic modification, play crucial role for condensing that part. Also after that, the condensation disappears. But when we are talking about condensation of a particular allele of a gene for a longer time period, for the whole life of a particular organism, then also this methylation and acetylation play crucial role. Like earlier in last class we talked about H3K9, H327, H317 methylation and acetylation process. Okay, so this LNC RNA model is quite important. So LNC RNA helps in genomic imprinting. This is true. So here is your next question. Imprinted genes are highly correlated to parental origin. Is it true or false? I think everyone can answer this question, as we have discussed about genetic imprinting here so many times. Try to answer this question. Okay, let's see. As I got the answer true, very good. That means you understood completely. That is true. Okay, imprinted genes are highly correlated to parental origin. So this is quite a description about genetic imprinting. In certain cases, the phenotype, okay, phenotype, you understand, one is genotype, another one is phenotype. Phenotype is which is expressed and which is the which shows the appearance of that particular organism, which genes can be we observe from outside. In certain cases, the behavior or phenotype conferred by an allele depends on whether that allele is inherited from mother or father. Okay, the basis of this parent of origin phenotypic variation that is most well characterized is genomic imprinting. Phenotype, we can call it as a behavior of that particular organism. Okay, outside behavior. And the essential role of imprinted genes include growth and development of fetus, postnatal behavior and metabolism. So this postnatal behavior uh, of animals generally is highly influenced by genetic imprinting. Okay, you will get very good examples of genetic imprinting for different types of animals. Okay, and metabolism. Imprinted genes may be either ubiquitously imprinted, that means imprinted throughout the whole body of that organism, that means it is ubiquitously imprinted or suppressed for all the cells, or exhibit tissue specific or temporal specific imprinting patterns. Got it? Sometimes it shows tissue specific, like for muscle tissue it is showing the imprinting, but for cardiac tissue it is not showing the imprinting process. Understood? They are located throughout the genome in approximately 1 MB cluster, and they expressed exclusively from maternally or paternally inherited chromosomes and under the control of a discrete imprinting control region, that is ICR. These imprinted genes must be marked with their parental origin so that the correct allele specific expression patterns are observed in somatic tissues. So now you understand the imprinting, how it is affects. So this is the main difference between genomic imprinting and doses compensation. So when we are talking about doses compensation, our mind will lead towards the bar body formation. That means one X chromosome will get suppressed or condensed to balance out the products of the two X chromosomes in the male and female, and this X chromosome get condensed to produce bar body in case of females. Okay, but when we are talking about genetic imprinting, we are talking about somatic cells, not sex chromosomes. We are talking about somatic chromosomes. Okay, and that's why it regulates the phenotype, appearance, behavior of the particular organism. Understood? So imprinting is related to somatic chromosome. Got it? These paternal specific marks must be stable, heritable, so that imprinting is maintained throughout the development. And this shRNA, that is small heterochromatic RNA, help in regulating or maintaining the imprinting process throughout the development of the particular organism, and it is essential. Erasable, so that imprints can be reset when embryonic germ cells are being reprogrammed during germ cell migration or differentiation. Okay, so this somatic imprinting, they must be erasable. It is not, this should not be like this, they are transferring from one generation to another generation. Then one particular organism will have the maternal chromosome expressed for 10 to 20 generation. It will not, it should not happen. Okay, when it is moving for germ cells. Okay, germ cells, you understand, that is the which will produce the ovum and sperms. The germ cells are being reprogrammed during germ cell migration and differentiation. This should be erasable. Okay, and stable and heritable. There are 2 points. Got it? So here is it about whole description about genomic imprinting. We talked about it earlier, but it is showing, it has some important points which I thought we should discuss. So it is about all about genomic imprinting. Okay, fine. Let's move to next question. Which of the following RNAs do not contribute in human diseases? Okay, miRNA, siRNA, rRNA, or LNC RNA. Which of the following RNA do not contribute in human diseases? Think about it a little, you can answer. Okay, let's see. Okay, so I got two answers, that is B and C. Some are siRNA or rRNA. So here is an answer, that is siRNA. Why is it? Can you tell me? Why is it siRNA which do not have or do not contribute in human diseases? Your answer is here. Okay, siRNAs in most cases originate from outer source, not from your host genome. Got it? That's why they don't contribute inside the host disease. Okay. Also, this small interfering RNA is a class of double stranded RNA, that is 20 to 24 base pair, that acts in the RNA interference pathway by interfering with the expression of genes with complementary nucleotide sequence to that siRNA, inducing mRNA degradation. We already talked about the function or mechanism of acting mechanism of siRNA, but let's discuss it again so that it will be helpful to everyone. See, this is a double stranded RNA or a shRNA, that is small heterochromatic RNA, which will be followed by dicer, that is one endoribonucleus. This is siRNA duplex. Okay, this is siRNA, it has two strands, that is 2 strands stands for the siRNA only. There is one particular enzyme AGO, which help in the formation of RISC complex. Okay, we talked about RISC, that is RNA induced inhibitor complex. So this RISC will followed by, this is a multi-protein complex. Okay, so RISC will followed by siRNA mRNA complex. How is it happen? Because the function of RISC is to take one strand of siRNA and find it complementary strand. Okay, so now this RISC factor took one strand of siRNA and got to know about this mRNA, which has its complementary base pairing. Now you got it, where there is a complementary strand is present. This is a double stranded structure, which is this is your siRNA, the above one. Okay, I am just making a star here. So this is one siRNA and this is the miRNA, sorry, mRNA. So this double stranded structure will followed by sliced mRNA by the enzyme RNase. RNase enzyme will cut the double stranded RNA structure, followed by silencing of this particular gene. Okay, so this is the process of or acting mechanism of small interfering RNA, that is siRNA, and they, as the function you can see, they just form a duplex followed by a double stranded RNA that will followed by the cleavage. So they don't contribute towards the any human diseases. Got it? See, let's move to another question. Homeosis refers to bone formation with wrong structure, formation with broken structure, or formation with big structure, or formation with malformed structure. Okay, so you guessed it correct. This is formation with malformed structure. Okay, so what is the meaning of homeostasis? This is homeosis. Now tell me, what is homeostasis? Anyone? What is homeostasis? Yes. Yes, very good. Balance. The balance between different body systems is called homeostasis. Okay, so here is the next question. Non-coding RNA is not involved in. Now we talked about non-coding RNA in different aspects. So try to answer this. Is not involved in development, differentiation, transcriptional regulation, or imprinting. Sorry, transcription elongation. Which one is it? Non-coding RNA is not involved in. Okay, so you answered it correct. Non-coding RNA not involved in transcription elongation. Okay, it has important role in transcriptional regulation, but not in elongation. Elongation, there are number of enzymes, proteins are there, but non-coding RNAs do not involve in transcription elongation. Okay, and it has important role in development, as we just talked about genetic imprinting, how it or this dicer molecule has important role in cellular development, differentiation, also imprinting. We talked about just now that long non-coding RNA has a crucial role in genetic imprinting. So it has almost uh, it is essential for each and every function inside the cell, but not the transcription elongation. Okay, so next, dicer acts in the nucleus nucleus nucleolus, cytoplasm, or nuclear pore complex. Which one is it? Okay, let's see. So you say cytoplasm. So it is correct. Okay. So dicer is a multi-domain endonuclease that belongs to the fourth class of type 3 ribonuclease or RNase 3. Fine. A dicer is encoded by a single gene in vertebrates and C. elegans, although it can be present in more paralogs in other species. Got it? So, dicer plays a pivotal role in the initiation of RNA silencing by recognizing double-stranded RNAs. Fine. So, here comes the role of dicer. It recognizes the double-stranded RNA and cleaving them into small RNAs using its RNase 3 like double stranded RNA specific nuclease activity. Small RNAs are largely classified into two groups, that is small interfering RNA and microRNA. So dicer has important role in formation of this siRNA and miRNA. Okay. And you can see here the schematic represent where the gene, this is a host gene followed by pre-miRNA or pre-mRNA or similar, followed by spliceosome. Spliceosome is a RNP complex which helps in splicing mechanism where introns have been removed. Okay, followed by the pre-miRNA. Okay, this leads to production of pre-miRNA. Now all this happens inside the nucleus. As you know, splicing mechanism is the important role of post transcriptional modification in nucleus, and this pre-miRNA move to cytoplasm. Okay, or cytosol. Here dicer, this is a which we can call it as double stranded RNA recognizing enzyme. It forms the miRNA and miRNA duplex and then act as a nuclease that cut the double stranded base pairing to produce one miRNA. When this miRNA interacts with the protein, it will be called miRNP. Okay, miRNA protein complex. Got it? Or ribonucleoprotein complex. Fine. So here it is the function of dicer, because we already talked about dicer and RISC factor a lot of times, so I thought this is important to you to understand what is the function of dicer and what is it. Okay, so I think it is clear to everyone that is it is basically a enzyme, or we can call it as a endoribonuclease, which help in this miRNA and siRNA formation from the double stranded RNA structure, and it recognizes and cleaves the double stranded structure. Fine. So here is your next question. RNAi or RNA interference was first discovered in Zebrafish, Saccharomyces, S. pombe or C. elegans. Okay, let's discuss. So this RNA interference was first discovered in C. elegans. Okay, you answered it correct. So how this was discovered? By the injection of a double-stranded RNA into the C. elegans resulted in the silencing of a gene complementary to double-stranded RNA. And I think you know the mechanism how this got happened. Okay, because when there is a double stranded RNA incorporated into the cell of C. elegans, this will followed by so this double stranded RNA will complementary to the gene. Okay, then it will form a double helical structure with the target gene. Fine. Then your dicer comes into picture where it will recognize its, then RISC factor will be there which will use this particular strand, and RNase enzyme will cleave the double stranded base pairing between your target gene and complementary RNA. Fine. So this type of mechanism happens. This is A stands for a negative control without hybridization probe and normal pattern of endogenous MX3 RNA. This is your normal pattern, and then injected with antisense RNA. We just talked about the mechanism of antisense RNA, how this just silence a particular gene. Okay, then injected with a double stranded RNA. Now you can see, if we just inject this particular gene C. elegans with double stranded RNA, this gene got silenced completely. Okay, in case of antisense RNA, we can observe a lesser dye or lesser interaction, but here lesser expression basically. But in case of when we inject a double stranded RNA, this will completely silence the particular gene which we want to suppress. Okay, MAX3 RNA. Got it? So this is the mechanism how it got discovered. So this is the last question of today's session. So hot air, the HOTAIR is a structural RNA, coding RNA, non-coding RNA, or sponge RNA. Which one is it? So I see you have answered C, that is non-coding RNA, and it is correct. Okay. So today we discussed about the mechanism of silencing process by small heterochromatic RNA, which helps in transcriptional regulation, translational regulation, also protein modulation, protein function modulation. Okay. After that we talked about RNAi, that is RNA interference, and the working mechanism of microRNA, that is miRNA, and siRNA, that is small interfering RNA. Then followed by your genetic imprinting in description and all about the different types of non-coding RNAs, how it regulates different gene functions. Okay, so this is all about today. If you have any doubt regarding any of this question, then you can ask me. Okay, then see you on next class on next Monday. Thank you everyone.